Welcome to our second part of our discussion of cell organelles. We're going to start this discussion with the rough and smooth endoplasmic reticulum. The endoplasmic reticulum is basically sheets and sheets of cell membranes, and it is a transport system and helps to transport proteins and hormones. There's two types of ER. There's the rough ER, which is covered with ribosomes, and the smooth ER, which does not have any ribosomes, and they both have very different functions in the cell. The rough ER, like I said, has ribosomes attached to it. It's usually connected with the nuclear membrane, and the reason for that is their functions are very much related. The ribosomes that sit on the rough ER make proteins that are to be exported from the cell, and once those proteins are made, they put them into the ER. So the ER doesn't make proteins, it transports the proteins that are made by the ribosome. Sometimes before transporting the proteins, they modify the proteins and then send them off in a vesicle to the Golgi body. Here you see a close-up of a ribosome. So here's the ribosome, and it is creating a protein. And as the protein gets longer and longer and longer, it puts it into the rough ER. And when it is finished, the final protein will then be packaged off in a vesicle and sent to the Golgi body. Here's the rough ER. You see it's very easy to recognize because of all of the dots on top of these kind of tubes. The smooth ER, on the other hand, is not attached to ribosomes, and that's because it doesn't have anything to do with proteins. What the smooth ER does is it makes or fat, and it makes steroids, which is a type of fat that you find in hormones like estrogen or testosterone. And the other thing that is very unique about the smooth ER is that it detoxifies harmful wastes. And that's why we find a lot of smooth ER in the liver. We also find a lot of smooth ER anywhere that you're making hormones, and those are called glands. Here's a picture of smooth ER. See, it looks quite different, but you can still see these tubes. Next, we'll talk about the Golgi body. Seen here in the picture. And the Golgi body is sometimes referred to as flattened pancakes. You'll see it's also tubes, basically flattened membranes. You think of a vesicle that's just been flattened like a balloon. That's basically what the Golgi is. And the Golgi is easy to recognize because there are always many vesicles on either side of the Golgi. And the reason for that is because the vesicles are bringing towards the Golgi proteins and hormones, and then the Golgi, once it's modified those proteins and hormones, is sending those vesicles out to the plasma membrane. So what is the job of the Golgi? The Golgi receives proteins and hormones from the smooth and rough ER, it modifies them, and it stores them until they're ready to be sent outside of the cell. The Golgi body also makes lysosomes. It's a great picture of a Golgi body, and you can see those vesicles here joining the Golgi, and then you can see here the vesicles that are pinching off of the Golgi and heading towards the plasma membrane, and showing the Golgi with the vesicles pinching off. This is a great animation of what happens when the vesicle reaches the plasma membrane. So the vesicle comes towards the plasma membrane, and all of its contents undergo exocytosis to leave the membrane. So I guess next we'll have to talk about vesicles. Vesicles are basically just storage sacs. They're small. They are formed when things come into the membrane by endocytosis, and they're formed at the ER, smooth and rough, and at the Golgi body when the cell is trying to send something outside the membrane. And they are used to transport things around the cell. They transport all sorts of things, from food to water, proteins, hormones, waste, old cell parts, if you look at a picture, you can see that it's basically just a piece of the cell membrane. You'll see the double layer of the phospholipids. The contents would be stored here in the middle. You can see the electron micrograph pictures. A vacuole is basically just a larger vesicle, usually formed when the cell is bringing larger things into the cell. A lysosome is a specialized type of vesicle. Again, like I said earlier, it's made in the Golgi. And a lysosome has special enzymes inside them. Those enzymes are called hydrolytic enzymes because they rip molecules apart. Sometimes lysosomes are known as suicide sacs because they have been known to attach to things and then cause them to explode. A great example of that is during development when a frog tadpole is turning into a frog. And the molecules that make up the tail of the fish-like tadpole get transformed into the molecules that are needed to make up the feet of the frog. Lysosomes digest the contents of food vacuoles and vesicles, and they do that with a process that's called hydrolysis. They also destroy old cell parts, and one of the places you'll see a lot of lysosomes working is in white blood cells. And white blood cells take bacteria into their bodies by endocytosis, and the lysosomes inside the white blood cell will then digest that bacteria. Here's an example of an electron micrograph of a lysosome, and the reason you know it's a lysosome is because it's dark. And the dark contents are representing those enzymes. The cytoskeleton gives the cell its shape. It anchors and supports the cell organelles. So if the shape of the animal cell is, for instance, in the reproductive tract of the female, and it has a rectangular shape, the cytoskeleton inside the cell membrane will help that cell maintain its appropriate shape. The cytoskeleton is also found within the cell, everywhere within the cell, and it acts as a transport system, almost like a subway or a monorail. And organelles, if they want to move around the cell, they have to move along this cytoskeleton. There's two parts to the cytoskeleton. There are the microtubules, which are quite large in comparison to the microfilaments. So microtubules are made of a protein called tubulin. It's a cylinder shape, and they make up three different things in the cytoskeleton. They make up the cilia and help anchor the cilia to the cell. They make the flagella and do the same thing. And they also make centrioles, which you would have learned about in your discussions of mitosis in grade 9. So the flagella in the human body, the flagella is only, whereas cilia, these short hair-like projections, are seen in different places in the human. And the cilia create a current outside the cell, and they help to sweep things or move things along. The flagella actually helps to transport the cell around when the cell needs to move. And the way that they move is this tubulin protein, and the tubulin protein is in pairs of two, and those two pairs slide against each other, and that's what creates the movement or the whipping motion of the cilia and flagella. The cilia are short, the flagella are long. Here's a picture of what the cilia would look like in the trachea, in your lungs, as they're sweeping dirty mucus out of your lungs to your throat. And of course, here's the sperm cell, the only place in the human body where we see flagella. Here's a cross-section of a cilia, and you can very clearly see these tubulin proteins in pairs of two. Microtubules also make centrioles, which are very clearly seen as a star-shaped organelle, and they have tubulin proteins in groups of threes. And basically what a centriole does is during cell replication, they grow these spindle fibers out of them, and the spindle fibers will attach to chromosomes and pull them apart from each other. So they attach to chromosomes during mitosis. And we only find centrioles in animal cells. Here's an electron micrograph picture. Here, it's not a very clear picture, but that is a centriole. And we'll just watch the process of mitosis. So there you've got your red centrioles growing spindle fibers, and spindle fibers pull those chromosomes apart so the cell can pinch off to make two new daughter cells. Again, the centriole replicates, becomes two centrioles, they grow spindle fibers, and pull the chromosomes apart. Here's a picture in an actual cell of that process. You can see here the spindle fibers and the centrioles, and the chromosomes here are being pulled apart. You can see that here, being pulled apart out of the pole of the cell, and ultimately you end up with DNA in each of your two new daughter cells. Microfilaments are also part of the cytoskeleton. They're a lot smaller. They're not made of tubulin, they're made of a different type of protein. And there's two types of proteins. They're called actin and myosin. And these are the really cool parts of the cell that help organelles move around the cytoplasm. If you look at a close-up picture of a cell using a different type of technology, you'll see all of these cytoskeleton. And again, here we have microtubules that make cilia, flagella, and centrioles. And then you've got your microfilaments, which help things move around the cell. And lastly, our cytoplasm. Cytoplasm is a watery, jelly-like fluid that's inside the cell. All of the organelles sit inside this fluid, and inside the fluid there are water, of course, salts, proteins, enzymes, other organic molecules that the cell uses to do everything it needs to do. Without cytoplasm, you wouldn't have enough water to make all of the reactions in your cell happen. So the cytoplasm is very important for reactions. And of course, it also helps to hold the shape of the cell. You can see in this picture the clear part of a plant cell is the cytoplasm. I hope you've learned a lot about organelles. Make sure you prepare those hot questions and we'll discuss it next class.